pretty straightforward.
Yeah, it's pretty.
I think you can go right to the textbook, and in that handout, so let's pull open that handout. That's this one. You can see my screen, correct?
Yeah.
Okay. So it gives you the page numbers, so 244 to 249. So they're there for you where each of these sections are. And so this is section 10. So let's just double check and just make sure that you can find it in the chapter. I forgot now what the page was. I want to say 246.
Like 244 to 249 or something.
Yeah, there you go. So there's the cell cycle stuff. And here's the part on the chromosomes.
Yeah, I think
so mitosis, cytokinesis. So yeah, you're basically just using the chapter, reading for information. So if you had two windows open at the same time, you should be able to get the information for that. And then it's an Edulastic. I forget how many questions, probably like 14 or 15 questions. You know, I can guarantee 15, 16, 17, 18, those are letters because it's really easy to do in Edulastic. I think the phases of mitosis are on there. Oh, there's quite a bit here. So it's pretty much a reinforcement of what I went over with you 2 days ago.
Yeah, I think I'm starting to get it a little bit more.
How about, I'm just going to go to it from here. How about this activity that you did yesterday?
For the most part, they were okay, but there were some, I don't know which ones they were, but I think they were towards the end. Those
okay. Let me just look at my key. So the beginning part is pretty straightforward. Let's just get this. And go to red. I'll go through. All right, so I'm just going from here. So you should, if you had, this was a C, this was a G, this is T and A, AT, AT, GC, CG, and the last one is TA. That's pretty much the culmination of these questions. It's just breaking apart all of these. Really here, the idea is for you to walk away with understanding that there's only four things, four nitrogenous bases, and the combination of those bases are what make up your genes. And the way DNA makes a copy of itself is, number one, there's only four things it has to worry about. So that's pretty cool. You don't have a lot of different chemicals to worry about. The other thing is it uses what we call semi-conservative replication. And that's really important to understand. Does that mean anything to you, or is that something that you're not comfortable with?
I think I get it.
All right, what does that mean? I put you on the spot.
Okay, maybe explain it one more time.
So semi-conservative replication, when we're talking about that with DNA, because that's one of the reasons why DNA does such a good job of making a copy of itself, is it uses this semi-conservative replication process. So what does semi-conservative mean with regard to DNA?
Isn't it like half of, that's what they said in the text. It was like semi is half, and then the conservative part, I don't remember that part.
Yeah, no, hey, that's pretty good. Let me tell you. All right, so let's see. Let's do, what would be the easiest way to do this? Okay, let's do it like this. We'll stick with red, and we'll just make this a little bit bigger, and it'll make sense to you. So imagine we have that strand of DNA. You know what? Just give me a second here. So the lines go straight. Nice little feature that it has. All right. So we have this strand of DNA, and it has these runs that go across. Does that make sense?
Yeah.
Okay, and just like in the picture here. So let's just draw them. We're going to end up erasing them in a second anyways, or erasing half of them. So it has these runs. It should have gone all the way across. What eventually happens is a chemical comes in, and it basically breaks all of this in half. It might straighten that out. It did. It's going to break that in half. And so what you end up with, so I'm going to erase that. And we'll go back to this. We'll get rid of this. is you're going to end up with one run here, one here. We'll do 4. Yeah, we'll go 5. We've got one down here. And these are all broken. So this picture here is basically representing what's happening between these. This strand is going this way, and this strand is going to go this way. So it's splitting in half. Now, in the middle, though, and what we're going to do here is let's extend this. Let's extend this like this, and let's do a couple more. And we'll extend this one, because that, I think, really is what, oops, 2, 3, I got to match them up. Perfect. All right, now we'll change the color. Let's see, blue's good. So now on the other side, if you remember, DNA gets replicated in what they call anti-parallel. So one end is going to go in this direction, and the other end is going to be replicated in this direction. So that if you were taking and putting cones down on the football field, you'd start at one end of the end zone, I would start at the other, and we would meet in the middle. Otherwise, if we both start at the same end, we might get caught up in each other. So sounds like an actual great way to be super efficient, so that's one of the tricks that DNA does. The other thing is what the semi-conservative is as this gets replicated, it's going to replicate here, here, here. I'm not going to put all the letters because you see what the letters are going to end up being. And then on this side as well, it gets replicated and replicated and replicated. Now, if you envision this, we could make this like visionary thing that in the center where you and I collide is kind of like where the centromere is in a chromosome. So now you're kind of getting the idea of when we draw chromosomes, we draw them like this because there's the original and the duplicated copy. But here's what's cool about it. Each half now, the original, so you got to remember this original chromosome, original DNA looked like this. Okay, now we've got one strand that has become one, but 2 strands. And each strand has retained, this is important, each strand has retained the red side. So semi-conserve, I've retained half conserve in the copy. So the original strand of DNA, when it makes its copy, it keeps in the copy 1/2 of the original DNA. So if you think about it, here's that 1/2, it breaks apart, and now it becomes part of the original copy. And the original copy still has it, there's the duplicate, but there is 1/2 of the original copy on this side, and there's 1/2 of the original copy on that side. Does that make sense?
Yeah, now I think I got it.
I mean, I did it in a much wordier way. You didn't have to do it that way, but that's what semi-conserve is. You're keeping 1/2 of the original strand when you make the copy. So if I'm keeping 1/2, it's like taking, it would be like making a photocopy and then only photocopying half of the page and putting the other half of the page right up to it. So if I lined them up, they'd always be right. Every copy would be perfect because it'd have to line up to the 1/2 that's the original. And that's just an awesome way. Between that and the DNA being replicated in opposite directions, that's just another way to make sure that every time you make a copy, the copy is correct. Because in the case of DNA, if you put a computer together and you're missing a part or you put the part in the wrong spot, the computer's not going to work correctly. In which case, that cell made up of that DNA won't work correctly. That's basically what cancer is. Cancer is, go ahead.
They're just right across from each other. One doesn't just like leave, right? They just stay across, but they're separated.
You mean this is what it, like right here, this is what it should look like?
Yeah.
So, okay, so now this is what would happen, which is, I'm glad you asked that. So this process that you just saw right here is what's happening in this stage of mitosis. So what just happened there is we have here, this will make sense. Why don't we, let's do this. Perfect. All right, so what you basically had was you have this cell, and we'll say that this cell is in the the G1 phase. So it's growing, it's doing all its stuff. I'm not going to put the nucleus in there. I'm just going to draw the DNA in there. So it's doing all its stuff. And the DNA is all inside the nucleus like that. Does that make sense so far?
Yeah.
Okay, so now that cell is going to grow old. Okay, so I'm going to go to here. So it's going to grow old. Actually, you know what the best place? I don't think I have them are the notes. That's okay. We could still do it this way. So you're right here. This is where we're living. If I could get this thing to work. So we're right here, and we're going in this direction. And this is where we are, G1. So this picture that I'm drawing here is G1. And we're going to move, I can move this over. Can I? That'd be cool. Okay. Then what happens is you get from G1, and then you're going to get to a point where the cell starts to grow either too big, or it's just not efficient, or it's just not doing its job as well as it should. It's getting old, like me. So now it says, all right, I need to duplicate my DNA. That's what you just saw. So what ends up happening is, we'll just go ahead and we'll draw another circle here. So what ends up happening, right, perfect. You end up with, now remember, this DNA is really broken up into chromosomes. So now let's just make believe for a second that the DNA kind of untangles itself. And we've got, in this cell, we've got a chromosome that looks like this, and we've got another chromosome that's a little smaller. So this particular cell only has two chromosomes, actually 4 chromosomes, but 2 pairs of chromosomes, meaning it has 1 chromosome from mom and 1 chromosome from dad. We're going to use the red for mom. And let's go to, let's do blue. Doesn't really matter. It's just 2 different colors. And this represents the other chromosome, and they don't have to be next to each other. Just give me a second to make sure there's not someone in the trying to get in. Now Aiden's here. Hi, Aiden. How are you? I'll take that as a good. All right. So what you just saw about this DNA semi-conservative replication is this strand of DNA floats around in your body, in your cell, and it communicates with the corresponding strand of DNA from mom. So you have half the DNA from dad, and you have half the DNA from mom. And it makes it a little bit better to understand this. If you envision, you remember those letters that we just did? So let's say A, and we had T, and we have C, and we have G. Well, in that DNA, the other side says T, A, C, and C. I guess I had 2 G's. So this right here may represent that span of DNA. And that whole chromosome, and that chromosome will be made up of thousands of little letter combinations. But on your chromosomes, you actually have these letter sequences that actually code for your genes, and they basically tell your DNA when to stop and when to start. And between each of those stops and start points represents an actual segment of letters that corresponds to a trait in your body. So your eye color may be on chromosome number 1. So we'll just make believe that this is chromosome number 1. And there are 2 halves to chromosome number 1. There's the half from that you got from dad, and there's the half you got from mom. And on that chromosome, on both, at each exact same location, there are little segments of DNA that code for particular traits. And we'll make believe that this first segment is eye color, and it has so many letters on it. Are you following so far?
Yes.
Okay, so now we need to take this cell and we need to reproduce that. Okay, so if you have eye damage, and eye color is not the best because it's not a cell that you damage. And I don't want to use skin color because then people will get all weird with me with skin color and stuff, but go with the eye color. So let's say you have damage to your eye. If you have damage to your eye and you want that to be repaired, you better make sure that the cells that repair it are exactly the same. They're no better, they're no worse. You got to think Mother Nature has refined that cell to be perfectly functioning. So it has to be an exact copy. It can't have a letter in a different order. Otherwise, it may make your eye polka dotted in that spot or something like that. So it has to be exact. So what you essentially have is so now visualize what we just did. You basically have that big ladder that looks like this. And this is your DNA. 1, 2, 3, 4. 2, 3, 4. What I am drawing is a neater version of this. And on this side, we've got, let's make sure I do this right. I want to just make sure I do this. Okay, we have an A and a T. And we have a T. You see what I'm doing, right?
Yeah, you're making that ladder form, right?
I'm making that ladder again. G, and we have a C, and we have a C. Okay? Now, what's going to happen is this chromosome, just mom's half. Now, we're not even worrying about dad's half. We're just worrying about the half that is mom, because I got to make sure that the new cell gets that half. So now what I'm going to do, and the whole line may erase. I may have to go through this again. Now I'm going to break that cell. No, I should keep saying cell. I'm going to break the DNA. And so now my line only goes halfway. And this one goes this, like this. So I've basically taken this chromosome, which really looks like this, like a ladder. It really looks with the 2 letters on both sides. I've now taken it and I split it, and now we'll use the other color. I shouldn't really use red, but I'm going to use, because that may be confusing. Let's just use a different shade of blue. And now one end of the letters, we'll just call them letters, are going to come in this direction, and the other end is going to come in this direction. And we're going to make, on this end, we're going to put an A, and we're going to put a whole new strand. Made it a little too. We're going to put a whole new strand right here, and we're going to connect them. And that strand is going to be a T, an A, a C, and a C. And then over here, this is going to make a new strand. And it's going to be an A, T, a G, and a G. What you just did is you kept one half of the mom's chromosome and the other half. Okay, you see what I mean? You kept the original. So now, if this, so now what you have is we've just gone from this picture to now this picture, and we'll just scroll down a little, where you've taken that chromosome, and we'll go to blue, and you've done this. Oh. I apologize. Go backwards. We'll do this. And we've done this. And then just because I used a different color, actually, you know what? We'll stick with the same color. We made a duplicate copy. So what we have is we have this, which really, if you zoom in, is a strand of chromosome that has 2 letters on each side. And then that strand of chromosome has to duplicate so that the new cell has the exact same. So in order to do that, mom's chromosome that you inherited needs to make an exact copy. And so that chromosome makes an exact copy. Originally, it was ATTAGCGC, and the dark blue represents the original portion of the chromosome. Now, when it goes to make the copy, each half has retained one portion of mom's original DNA plus the copy. And the other copy of the chromosome contains one half of mom's original DNA plus that piece there. So it now has duplicated and made 2 parts of mom's DNA. We've got the original which is now split in half, and half of the original is here, and the other half of the original is on this piece. Are you following that?
Yes.
Okay, so now the same thing is going to happen with the other chromosome number 1. So the other chromosome number 1 is the one you inherit from dad. And that chromosome number 1 looks like this. And we'll just say it is A, T. Totally different T, 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 A. There's no G's and C's in that chromosome that goes for that. I don't know why, but it still codes for the same stuff. That's what I'm making up, and I'm sticking to it. So now if we were to do the same thing, we need to now take this chromosome that looks like this, and we need to duplicate it so that the new cell gets exactly 1/2 of that exact. Because remember, you need both chromosome number 1s to make your traits work. If I have chromosome number 1 for mom and it has eye color on it, but there's no chromosome number 1 floating around in my cell from dad, that trait won't work. It needs both traits to work. They're actually called alleles. It's the reason why you did Punnett squares. What we're really doing here is the background behind what happens in a Punnett square. It's just that you guys don't learn that. You just learn that you put letters in boxes and they end up. Do you remember Punnett squares?
I don't
You put
remember.
the letters in the boxes. They look like this, and you're like capital R, lowercase r, like all that gene stuff.
I think like a little, but not really.
All right. Okay. So I don't want to lose you here. So let's go back. Okay, so we're not, without dragging through the process, what we're going to assume then is that this chromosome is really, if we zoom in, looks like this. And if it were to make a copy, it would end up looking like this in the cell, like this. And now it has its copy. It would have gone through this same process. It would have split into 2. And we would have had the same thing. 1, 2, 3, 4. 1, 2, 3, 4, and we had A, and T, and this one was T, 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 and A. And what would happen is a strand of copies would come in one direction, and we end up with the exact same thing, which is what this represents here. So we essentially end up with dad's chromosome and an exact copy. Now, here's the thing. I've got the chromosome that I had originally, and I've got this copy. But how do I get it so that it goes into the next cell? Because this cell is going to split in 2. In other words, if the cell were to split right now, let's just assume that the cell were to split. If that happens, that would not work. And here's why. Because remember, the original cell has 1 red and 1 blue. That's 1 chromosome from dad and 1 chromosome from mom. If this cell were to split in half the way these chromosomes are assembled or arranged right now, instead of each cell having 1 chromosome from mom and 1 chromosome from dad, each cell would have, in this case, the chromosome from dad in the copy, and the other cell would have the chromosome from mom in the copy. You see what I mean?
I think so. It's not like the exact same on both sides.
No, it's not the exact same. Think about it. If your original cell was one red strand and 1 blue strand, we're not even going to worry about these right now. 1 red strand and 1 blue strand. So that's your original cell. Well, I can't do it because I changed colors, but your original cell has a red strand and a blue strand. If this cell separates right now in the middle here, and this one becomes its own cell, and this one becomes its own cell, you're going to have a cell that has 2 red strands. That's not exactly the same DNA as the original. You see what I mean?
Yeah.
Okay, so, well, do you see how that's not the same as the original?
Yeah.
Okay, as long as you know that that's not the same as the original, because here's where I'm going with this. Because it's not the same as the original, your cell wouldn't function. So if your cells did that, they would not be viable. They would die. And I think that's a word that I think comes up in the activity that you were doing, or if not, it comes up on Friday. So what happens, which is actually kind of neat. So I'm going to just move up a little bit. So let's get rid of that squiggly line. Oop. I don't know if I went through this with you, but we'll do this. What actually happens is in there, and you'll see this on Friday, so I'm kind of teaching ahead. There are little structures that when a cell starts to get ready to actually divide, So let's kind of like, let's just make sure we know where we are right now in the cell cycle before we go any further. We are, Poggle. We are here in the cell cycle right now. We have synthesized our DNA. So in the life of a cell, we were born, we grew, we realized we were getting too old, and we duplicated our DNA in the assumption, anticipation that we will have to divide into 2 cells. So that's where our diagram lives right now. When it gets to the point where it has to actually divide into 2 cells, your cells will start to develop these little structures, and this will come up tomorrow, but they're called centrioles. I guess we could label them here. Centriole. And they will put out these long strands of stuff. Did I do this with you already or no?
No.
Good. This is good then. And what that means is these strands of stuff, another new term here, are called spindle fibers.
Oh yeah, this word spindle was on one of the questions.
Was awesome.
For today, I think.
Okay, good. So what these spindle fibers do is they move everything around. So now remember what we said. If the cell splits like this, we are not going to end up with what our original cell looks like. And let's make our original cell so we can see it. So it's nice. Let's go to our original cell, and we'll just put it there so that it makes perfect sense. Our original cell is going to have 1 red and 1 blue. If this thing splits, it's going to end up with 2 reds and 2 blues. So how does that work? All right. And we'll go this direction. We're all over the place here. This is great. Well, now we're blue. So we went from a pink cell to a blue cell, so we'll have to go with it. So now we'll do this freehand. All right. So let's put our centrioles here, and we'll put our spindle fibers out. What they're going to do is they're going to take those chromosomes and they're going to move them in such a way, and they're going to take them and they're going to place this one on top, and they're going to take the blue one and they're going to place it right there. I did okay. And now what happens is when the cell starts to pull apart. So now what'll happen is this half of the cell will pull in this direction, and this half of the cell will pull in this direction, and the cell will start to split, and it'll split nice and thin. Do this as best I can. It'll split right down there, and what you'll end up with then is you'll end up with 1 cell that has one blue, got that blue one. This one will have the other blue, and then this half of this red will be over here, and this half of this red will be over here. And now if you look, these 2 cells are identical to my original cell. So what I'm trying to point out to you is when the cell duplicates, what I was drawing with you before with all the letters and it going out and the semi-conservative. The semi-conservative part is what's happening in the synthesis phase. It's where the DNA is being duplicated, and the way DNA prevents itself from screwing that process up is that in each copy, it keeps 1/2 of the original strand of DNA. That ensures that it makes it very difficult to screw things up if you keep 1/2 of the original, because the parts have to line up. And then the other thing that I tried to show you here was that when cells begin to divide, there is a step, and I didn't even identify the step because I want you to discover it for yourself, but it's called metaphase. There are little structures that will move the chromosomes around and arrange them in such a way that allows them to split apart and form 2 identical cells. And so if your chromosomes were lined up in this like this, that would not work because you would not get the exact same as the original cell. What you would end up with, and let's put some terminology here, you would end up with the chromosome and its sister chromatid. Do you understand what I mean there? You would end up with the original dad chromosome and its copy, which is called a sister chromatid. And the other cell would end up with mom's original chromosome and the copy, which is called a sister chromatid. There's a term that you may or may not have, maybe in the homework for today. Is there a term called homologous chromosomes that came up?
What was it called?
Homologous?
I haven't seen that, but I haven't finished the whole thing.
Okay, homologous chromosomes are what we refer to when we say you have chromosome number 1, you have another chromosome number 1. And that chromosome number 1 matches with the other chromosome number 1. In other words, chromosome number 1 from dad and chromosome number 1 from mom are homologous because they share the same sequences of DNA that code for specific genes. That's why you can't have a gene work correctly if you only have one half of that chromosome. You need the pair. It's like having your left sneaker and your right sneaker. So here's a really good analogy that seems to work with kids. If you were just, and I wish I had a pair, like, let me see. Let's see if I could find something that would really help here. Pair of sneakers and laces. You're probably like, what is this guy looking up? Pair of sneakers and laces. Let's see what we get for images. Okay, so this will work. This will work. I don't think I'll be able to do much with the image. Let's go here. Let's copy image. And control V. Oh, perfect. Yes. Look at that. Super duper. Can I draw on this thing? Because if I can draw on this, that would be like totally cool. Oh, wow. Look at that. All right. So let's make this a little bit. Okay. What you got to think of is in your cells, your chromosomes are like a pair of sneakers. And one chromosome is, we'll say these pair of sneakers represent chromosome pair number 1. And you have a left foot, number 1, and you have a right foot. Actually, that was your right, and the other one's the left. Number 1. Making sense so far?
Yes.
Okay, you need the right foot and the left foot in order to make anything work. Each one of these shoelace holes here represents a particular gene in your body. The first one could be eye color. Second one could be whether your earlobe is attached or not. One of them could be whether you can make your nose go like this. One of them could be if you can roll your tongue. I'm just pointing out ones that are kind of cool. One of them could be, I think if you can do something like that, I think that's genetic. Now, and those all have thousands of little letters. Remember we talked about the letters A, T, C. Okay, so they have thousands of letters that make up a particular sequence. And that sequence of letters in that segment corresponds for a particular variation of that trait. So that segment that I said is eye color, maybe your mom has the segment of letters that codes for brown eye. That's what she has. Now your dad, you need the other half of the chromosome to make your eye turn on. You can't turn it on with just mom's chromosome. It needs the other half. So you need the other homologous chromosome. You need the other foot floating around. And on that, okay, that represents eye color. That's whether your ears are attached. The exact same order of these is matching with the exact same order on the other homologous. Homo means same. The prefix means same. That's why they call them homologous chromosomes. Are you following so far?
Yes. So they have to have the same on both sides.
Have to have the same on both. That's right.
Okay.
So now dad maybe has blue eyes because the 1,000 letters on that eyelet that make up eye color, his arrangement of letters codes for blue eyes. Now, when you get dad's blue eye gene and you get mom's brown eye gene, unfortunately, one of them is boss and the other one is wimp. And the bossy one is the one that's expressed in you. That means you may have brown eyes, but you may carry the variation for blue eyes. That's why when you have your own children, you may, unknown to you, that child may be blue-eyed simply because you, the same thing, you passed on your dad's half of the eye color gene and your baby didn't get mom's gene that you inherited. See what I mean? That's how come there's variation. Are you okay so far?
Yeah, that actually makes more sense now.
Okay, now here comes the good part. So now we talked about that ladder. That ladder, this ladder right here, represents this side, and this ladder represents this side. Okay, so far?
Yes.
Okay, now watch. It's going to blow your mind. Actually, I really should say, because I'm going to go, I think if I, that's not good. Okay, whoa. How much far back can I go? Okay, what I really, I mean, to make this really work, and it won't make a difference, is really this, I should say, represents over here, and it's on this side. That's fine. Okay, it doesn't really matter. Now, these bonds are exactly what these bonds represent. So now what happens is these bonds break apart, and now you end up with, so now this is where the chromosomes duplicate. If this is the order, and this strand out here represents, so remember, this is the dad chromosome, this is the mom chromosome, and now the chromosome split and has to make a duplicate of itself. So when it splits, what you're already seeing is a split that happened. This split happens, and whatever's on this side is duplicated here, so that this chromosome will end up being an exact copy of this. And this, when it becomes a duplicate, will be an exact copy of this. Are you following that, or did I lose you there? I may have lost you.
No, I think I'm following.
Yeah? So
Yeah.
that's okay. So like that's the piece that's really important to understand. It almost makes more sense if we talked about the pair of shoes and we just said that this part here wasn't there yet and that the pair of shoes were kind of together and then they split. So this is, the pair of shoes are split, and this portion here is the new strand being formed, and this portion here is the new strand being formed, and that's where you end up with your 2 DNA. Here's the original strand, and then we have one half of the strand is retained on this side of the sneaker. One half of the original strand is retained on the other side of the sneaker, and the new strand is put here that resembles what's over here, and this new strand goes here, which resembles what was on the other side. And that's what semi-conservative replication is. That's what the chromosomes are. And then they get rearranged, and we put one to one cell and one to the other cell. I hope that helped.